Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
Today we are looking at a drafted outline for a highly technical review article arguing that transitioning from transition metal catalysts to ketoreductases, or KREDs, is a mandatory survival strategy for cost-effective, high-purity API production.
Right, and jumping right in, the current division between process engineering and protein engineering obscures the central industrial tension between biological mildness and manufacturing concentration.
Okay, so you're looking at the overall structure right off the bat.
Exactly. I mean, when you analyze how this outline is structured, keeping solvent engineering in Section 3, cofactor economics in Section 4, and then putting protein engineering entirely in its own silo over in Section 5, it just fractures the narrative.
Yeah, it breaks it up too much.
Right. By isolating the biological limitations of KREDs from the physical process and the molecular solutions, the article temporarily loses its cohesive argument about scalability.
Because the reader has to bounce around?
Yeah, the reader is forced to jump from, the physical constraints of aqueous phase processing all the way to the economic hurdle of NADPH, and then only pages later do they read about how the enzyme itself is actually altered.
Which kind of misrepresents how those decisions are made in real life, right? Like during pharmaceutical scale-up?
Exactly. That structural fragmentation fundamentally misrepresents reality. In the actual manufacturing landscape, you do not solve a solubility problem in a vacuum, and then, just hand it off to a geneticist.
Right. It's highly collaborative. So how should they fix that?
The suggestion here is to restructure these sections into a unified engineering the catalyst and the process supersection.
Oh, I like that. A supersection.
Yeah, this revised format should pair each physical limitation directly with its corresponding dual solution. The text needs to explicitly weave together how altering the enzyme's environment and altering the enzyme's DNA are basically two sides of the exact same coin.
Let me throw an analogy at you just to see if I'm tracking.
Go for it.
It is like trying to explain how a high-performance car works by describing the engine and then completely separately describing the chassis.
Yes, exactly.
Like you cannot upgrade the engine via protein engineering without reinforcing the chassis through solvent and cofactor systems. They really have to be discussed together to make sense of the vehicle's top speed.
That is a perfect analogy. You cannot upgrade the engine's top speed without reinforcing the chassis to handle the torque. In this case, the chassis is the solvent environment and the cofactor recycling system. If you discuss them separately, the vehicle's performance metrics make zero sense.
So, looking at the hurdle of substrate solubility, how does this play out in the draft?
Well, moving reactions away from toxic volatile organic compounds like DMF or DCM and into benign aqueous buffers aligns perfectly with the 12 principles of green chemistry.
Right. Everyone wants to be green.
But aqueous buffers are notoriously terrible at dissolving bulky, highly lipophilic ketones.
So you hit a wall.
Yeah, and if you introduce, say, neat substrate loading or aggressive bio-based co-solvents to force that solubility in a massive 200,000-liter reactor, you introduce immense toxicity to the wild-type enzyme.
It just kills it.
Exactly. Therefore, the author must merge the discussion of co-solvents with the directed evolution workflows. The text has to demonstrate how design, build, test, learn cycles are deployed to explicitly mutate the KRED's active site and surface residues.
So the protein actually survives those exact non-natural industrial conditions.
Right, exactly. And building on that point, we have to look at how the draft handles the NADPH challenge.
Yeah, the cofactors?
The outline currently treats the reduced nicotinamide cofactor as sort of a biochemical footnote. Like just a necessary ingredient that is simply too expensive to use stoichiometrically.
Which, it is expensive.
It is, but what if we looked at it from the perspective of the cofactor being a solved engineering parameter? The text should group the NADPH limitation directly alongside the deployment of glucose dehydrogenase, or GDH-coupled systems.
Grouping them together changes the narrative entirely.
It really does. By bringing the GDH regeneration strategy into this unified section, the author delivers a crucial conceptual insight. Transitioning to green biocatalysis doesn't eliminate process complexity, it simply relocates it.
Wow, that's a great way to phrase it. It relocates the complexity.
Yeah, because instead of utilizing the mechanical brute force of high-pressure hydrogen gas over a palladium catalyst, you are engineering a delicate thermodynamic loop.
Where the GDH oxidizes the glucose.
Exactly. It oxidizes glucose specifically to regenerate the NADPH that the KRED consumes. And that localized catalytic network must be engineered for extreme stability.
And rigorous byproduct management, right? Like dealing with the accumulation of gluconic acid.
You got it. The tension between biological mildness and industrial concentration is where the science actually becomes interesting. Unifying these sections really captures that friction.
That makes total sense. So once we've established how process and protein engineering work together to create this highly efficient system, we need to look at how we are actually proving this efficiency to the reader.
Right. And if we agree that process and protein engineering are two sides of the same coin, then the metrics we use to measure their success cannot be siloed either.
Which brings us to the math.
Exactly. Isolating the E-factor and process mass intensity metrics into a standalone section dilutes their persuasive power by separating the theory of waste reduction from actual industrial execution.
It just leaves it hanging out there on its own.
Yeah, dedicating Section 6 entirely to abstract metrics like E-factor and energy efficiency creates a dry, theoretical lull in the pacing.
Oh, I can see that. It slows down the read.
Right. When the text says an E-factor drops from the tens or hundreds down to single digits, the impact is lost because it isn't immediately attached to a real-world active pharmaceutical ingredient.
So the reader is basically waiting around until Section 7 to see the actual industrial case studies.
Exactly, which lessens the impact of the KRED platform's ability to, eliminate protection and deprotection steps or completely bypass heavy metal scavenging.
Let me play devil's advocate for a second, though. Is there a risk that focusing solely on abstract E-factors might overlook what a pharma manufacturer actually cares about?
How do you mean?
Like a manufacturer isn't going to care about clean chemistry as a concept until they see exactly how it streamlined FDA and EMA approvals by reducing residual metal impurities in a specific blockbuster drug, right?
That is absolutely correct. And that's exactly why the standalone section doesn't work. The math only matters when it solves a specific logistical nightmare.
So what's the fix here?
The suggestion is to completely dissolve the standalone metrics of success section. Just get rid of it.
Okay, just scrap it entirely?
Scrap it as a standalone, yeah, and instead embed the E-factor, energy efficiency, and waste minimization data directly into the industrial case studies of Section 7. Make the metrics the dramatic stakes of the real-world stories.
Give me an example of how that would look in the text.
Sure. Let's look at the ipatasertib intermediate case study. Right now, the draft uses that drug solely to showcase KRED engineering at elevated substrate concentrations.
Right.
But they should use it to not only show the concentration engineering, but to explicitly showcase the dramatic E-factor drop simultaneously.
Like putting the numbers right next to the drug name?
Yes. Walk the reader through how the route redesign brought the E-factor from 105 in discovery down to just 7 at the production stage.
Wow. 105 to 7? That's massive.
It is. And, when discussing this in a university setting, I often emphasize that the E-factor in pharma is notoriously high. It's often 25 to 100 plus.
Yeah, it's wild how much waste is generated.
Right. So switching to a KRED-based system can often cut that waste by half in a single unit operation. By explaining why it was 105 initially, due to cryogenic cooling requirements, solvent extractions, multi-step protection sequences, that drop to 7 via a tailored KRED becomes a monumental industrial triumph rather than just a dry statistic.
It gives the number context, and I assume the same goes for the statins they mention?
Absolutely. When discussing the synthesis of statins like atorvastatin or antivirals like cimepravir, the authors should immediately highlight the shift to ambient temperatures.
Like what, 25 to 35 degrees Celsius?
Exactly. Contrast that with the cryogenic or high heat requirements of previous multi-step metal catalyst routes. When you put the efficiency metric right next to a globally recognized drug, the abstract concept translates into tangible operational savings.
Because when an executive looks at a transition metal catalyst like rhodium or ruthenium, they don't just see chemistry, they see a regulatory bottleneck, right?
Yes, exactly. They see the requirement for costly, labor-intensive scavenging steps to ensure the metal parts per million fall below stringent FDA and EMA thresholds.
So by embedding it, you're proving that biocatalysis shifts the burden from downstream cleanup to upstream genetic design.
Precisely. You are proving that enzymatic keto reduction is an enabling manufacturing philosophy.
Okay, I love that. Now that we've grounded the current industrial proofs in hard metrics, let's look at how the draft handles the future of this field, specifically regarding digitalization.
Right, and this is crucial. Treating AI and digitalization as a disconnected future frontier risks making the conclusion feel like an optimistic afterthought rather than a rigorous scientific projection.
An optimistic afterthought? So, like, it feels tacked on.
Yeah. Section 8 currently approaches AI-driven bioprocess optimization as a separate conceptual bucket. And while touching on digital twins and in silico screening is super timely, presenting it without firmly anchoring it to the physical limitations of the laboratory makes it sound like a magic bullet.
Ah, I see. It risks implying that AI can just replace the messy reality of wet lab validation.
Exactly. Which undercuts the deep synthetic organic chemistry expertise the author established in all the earlier sections.
That makes sense, but let me push back constructively here. Could the evidence also support a different conclusion?
What are you thinking?
For instance, if we lean too hard into predicting enzyme substrate fits before a single wet lab experiment is performed, do we risk alienating the traditional organic chemists reading this? The ones who know firsthand that an algorithm can't account for every solubility hurdle in a 200,000-liter tank?
That is exactly the danger. Framing it as a magic bullet is highly dangerous for this specific audience.
So how do we fix the angle?
Recalibrate the angle of the AI section to frame machine learning strictly as an accelerator for the design, build, test, learn cycles discussed earlier.
An accelerator, not a replacement.
Right. Explicitly highlight its limitations and its absolute reliance on physical chemistry. Emphasize that while in silico enzyme substrate mapping can predict fits and, compress the search space, it fundamentally cannot replace the wet lab test phase.
Especially when dealing with bulky or sterically hindered ketones.
Exactly, because those behave unpredictably in actual reactor conditions. Algorithms struggle significantly with the variables of mass transfer and fluid dynamics.
Right. Localized pH shifts, biphasic systems inside a massive impeller tank. The computer might say yes, but the tank says no.
Exactly. The computer calculates a perfect fit in a vacuum, but physical friction means only empirical testing can validate it.
And what about the digital twins they mention? How does that tie in?
Well, instead of treating the digital twin as an isolated buzzword, tie the concept of digital twins and fermentation directly back to the earlier point about E factors.
Oh, brilliant. Connect it back to the waste.
Right. The KRED enzyme is renewable, but fermenting the E. coli host requires massive resources: carbon sources, aeration, precise feeding.
So show how real-time data optimization ensures the production of the KRED enzymes themselves doesn't become a new source of process waste.
Exactly. That grounds the digital concept in the physical reality of the 2030 zero waste mandate. You want traditional organic chemists to view machine learning as a sophisticated tool to enhance their enantiomeric excess, not as a theoretical replacement that ignores physical scale-up.
That is a phenomenal pivot. Okay, so just to briefly recap our main takeaways for the author first, seamlessly blend the process and protein engineering sections to highlight the synergy of industrial scale-up. Second, embed the E-factor and efficiency metrics directly into the blockbuster case studies for maximum real-world impact. And finally, firmly tether the AI predictions to the rigorous demands of wet lab validation.
Beautifully summarized. We warmly invite the listener to revise their review article based on these structural and thematic alignments, and please submit the next iteration back to the critique for another round of feedback.
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Urnala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.